Thank you very much. Well, good evening, ladies and gentlemen. Wonderful, packed house. So let me say a few words about the speaker this evening. Philip Ball is a freelance writer. Previously, he was an editor at Nature for over 20 years. He writes regularly, both in the scientific and popular media, and has written many popular science books. I haven't read all of them, but I've read several of them, I promise. So some of his books, H2O, A Biography of Water, Bright Earth, The Invention of Color, The Music Instinct, Curiosity, How Science Became Interested in Everything. He won the 2005 Aventis Prize for science writing for his book Critical Mass. In fact, I think that was the first occasion I met Phil. I was sitting at his table. My wife and I were sitting at his table, so obviously we were cheering him on. Do you remember that? And then so we whooped with delight, as though somehow we could share and reflected glory when his name was announced as the winner. He's been awarded the American Chemical Society's Grady Stack Award for interpreting chemistry to the public. He was the inaugural recipient of the Lagrange Prize for communicating complexity science. He trained as a chemist at Oxford and as a physicist at Bristol. So this man is not a hack. This man is very, very smart. And he covers a wide area of science. He was one of the first people we sent the draft of our manuscript to, my book Life on the Edge, when we wanted some expert opinion, someone to cast their eye over it. Phil's latest book, by the way, is Invisible, The Dangerous Allure of the Unseen. He wrote the article in Nature to which that picture is associated in 2011, but as early as 2004, I think, 10 or so years ago, Phil wrote an article in Nature about how quantum mechanics plays a role in the action of enzymes. So this is essentially before even many in the world of science, let alone in the wider public, had even heard of the subject of quantum biology. So now that this field is really emerging and a lot of people are getting very excited about it, I think there's no better person to provide an introduction to the field than Philip Ball. Phil, over to you.
Thank you very much. Thank you, Jim. It's very flattering to be entrusted with this rather daunting task. I'll see how I get on. I guess like most trendy fads in science, quantum biology is actually rather old. And I want to suggest to you that it begins on the 15th of August, 1932, with this man. This is Niels Bohr, the Danish physicist. And he gave a talk at a rather unlikely-sounding meeting, the International Congress on Light Therapy in Copenhagen. And Bohr's title was Light and Life. And he offered some thoughts on what the new theory of quantum mechanics, which he had just helped to formulate in Copenhagen during the previous decade, what it had to say about biology, about life. Well, Bohr was the right man for this job. Not because he had any real understanding of the answer to that question, but because he wasn't someone to let ignorance get in the way of speculation. And I mean that in a good way, because when smart people like Bohr are prepared to stick their necks out and say something on a topic about which they know rather little, there's always a chance that they'll spout nonsense, but I think there's a better chance that they'll spark something valuable. And I'm trying to say that without looking in Jim's direction. In his talk, Bohr asked, and I quote, whether some fundamental traits are still missing in the analysis of natural phenomena before we reach an understanding of life on the basis of physical experience, by which he meant physics. But he left that suggestion rather vague, and it's not really what Bohr said that matters, but who was in the audience. Because Bohr had been very anxious to make sure that among the audience was a young German theoretical physicist named Max Delbruck, Here he is. And soon after returning from Copenhagen to Berlin, Delbruck decided that the kind of missing trait that Bohr had alluded to might be found by studying the physical and chemical nature of genetics. And by that, Delbruck didn't mean that physics might help to explain genetics, but rather that by studying genetics, he might uncover some new principle of physics. Now remember that at this stage, virtually nothing was known about the molecular origins of genetics. People had begun to understand how genes evolve in a population according to Darwinian principles, but they really had no idea what genes were. They didn't even know that they were associated with DNA. And in 1937, Delbruck went to America, where he began to investigate the genetics of bacterial viruses, since they were a very simple form of life. And this work eventually won him a Nobel Prize for his work on how viruses replicate. And Delbruck established the idea of genetics as an information science. It was all about transmitting information. He also helped to stimulate the interest of other physicists in biology, and one of them was this chap, Erwin Schrödinger, who in Dublin in 1944, after having fled the Nazi regime, he published an extremely influential little book called What is Life? And here, Schrödinger pondered the question of how an event at the atomic scale, a mutation in the molecular structure of a gene, how this, while being governed presumably by the laws of quantum mechanics, could potentially cause a profound and visible change in the structure of a living organism. And Schrödinger speculated that these genes must be something that he called an aperiodic crystal, that is, an arrangement of atoms that was highly specific, not random, but that didn't have a regularly repeating structure like that of a crystal. Now, another physicist lured into biology by Delbruck's work on replication was Francis Crick, who, of course, in 1953, with Jim Watson, showed that Schrödinger was right, that genes are made up of very specific sequences of molecular components along the double helix of DNA. According to the eminent molecular biologist Gunther Stent, Crick and Watson's discovery brought about the wedding of the informational school of Delbruck with the structural school of crystallographers who were looking at the atomic scale structures of biological molecules like DNA and proteins. And that, Stent says, made molecular biology what it is today. So there you have it. It wasn't just that physics created modern biology, it was specifically quantum physics that started it off. Well, that's all very satisfactory for smug physicists, but it doesn't say anything about Bohr's intuition that quantum physics has any actual role in biology. Now, of course, in a trivial sense, it must do, because as Bohr himself showed in the famous papers in 1913, where he laid out his theory of the atom, quantum mechanics can explain why it is that atoms bond together to form the molecules like proteins and DNA and RNA on which all life depends. So in this sense, quantum theory is at the root of everything, and we need it to explain everything. We need it to explain why I'm standing here talking to you tonight. Now, I know that quantum theory can seem scary or mysterious to some people who are not so familiar with it. And here, one is usually supposed to reassure you with Richard Feynman's famous quip that no one understands quantum mechanics anyway, and that if you think you do, then it shows you don't. Now, I think that that's true at the level that Feynman intended, but it doesn't mean that quantum theory is hard to understand. It's just hard to understand what it means. The challenge isn't so much in understanding what quantum theory says, it's in accepting that information. So let's see how we get on. It was known by the early 20th century that chemical bonding is all about the electrons that atoms contain. Ernest Rutherford had proposed that atoms are a little like tiny solar systems with this dense, very massive nucleus and electrons orbiting around it. And Bohr said that those electrons can only have specific energies and no others, and that they can therefore only adopt some orbits and not others. And they can jump from one orbit to another by absorbing or emitting energy, for example, by swallowing up or spitting out a photon of light with a particular wavelength, which is related to its energy. When electrons do this, they are said to make a quantum jump. And Bohr outlined how electrons can adopt quantized orbits that link two or more atomic nuclei and that will join them then together in a chemical bond. Here are some of the early sketches that Bohr made of that idea. Well, this quantum theory of atomic bonding has got a lot more sophisticated since then, and it now gives a very detailed and precise description of the shapes and the properties of molecules. It explains, for example, why grass is green and why blood is red, questions that John Donne asked in the 17th century. And they have these particular colors because the energies of the electrons quantum states in chlorophyll and in hemoglobin are what they are, so that they're the right energies to absorb red or blue or yellow or green light, whatever the case may be. So in this sense, molecular biology depends on quantum physics. But when we talk about quantum effects in nature and in science, we generally mean something different from this. Quantum mechanics is notorious for producing effects that defy everyday intuition. Particles can behave like waves. They can be in two places or in two states at once. They can pass through walls and apparently communicate with each other instantly over vast distances. Now, we tend to be insulated from actually experiencing these odd behaviors on the whole, either because they only appear in rather specialized conditions, such as at very low temperatures, or because their operation at the scale of single atoms and molecules becomes washed out when averaged away for objects at everyday scales that contain trillions upon trillions of atoms. This is why we can generally rely on the pre-quantum classical physics, classical mechanics of Galileo and Newton to explain and describe how things behave in the everyday world. Quantum biology explores the possibility that some of these strange quantum effects can happen in biological systems, where they might, in fact, turn out to be essential for the way the system does its job. Now, this seems, on the whole, to be very surprising and unexpected. If physicists want to study quantum effects, then very often they need extremely sophisticated equipment. They have to cool things down to maybe within a few degrees of absolute zero. They might have to create extreme vacuums greater than the, more empty that is, than the vacuum of interstellar space. They might have to take great pains to make sure the things aren't disturbed by the environment. Biology, in contrast, the world of the cell, is warm and wet and messy. Cells are little bags of countless different types of interacting molecules, all colliding and reacting. And expecting to encounter quantum effects here is a bit like expecting to be able to sit down and meditate peacefully in Waterloo Station in the middle of rush hour. And yet, it happens. There's good evidence that quantum effects like this do matter in biology, and I'm going to tell you about some of them. But I want to stress that to what extent they matter, whether they happen at all, and if they do, whether they're biologically relevant, this is something that's still being intensively debated. These effects seem to be well attested in some cases, they seem to be uncertain in others, and extremely speculative in others. Now, this is natural. Quantum biology is still in its infancy, and we can't yet say whether it amounts to just a ragbag of oddities and quirks of nature, or whether it's a profound aspect of how life happens. Well, let me start with waves. Wave-particle duality is probably one of the least helpful expressions in quantum theory, but what it strives to convey is that very small particles seem, in some experiments, to show the characteristics of particles with particular positions in space, let's say, and in other experiments to behave like waves, which are spread out through a large region of space. The particles of light called photons do this, and so do electrons. That's how electron microscopes work, by making use of the wave-like character of electrons. And so do atoms. Now, here's a picture of atoms. This is a real picture of atoms where each of these blobs is an individual atom. I mean, sure, they've been colorized and, tidied up to look this way, but really that's what you're seeing here. Atoms arranged on the surface of something like a metal into these patterns. And yet here is what happens if you fire atoms through two narrow slits in a screen. First of all, you see that the atoms seem to appear on the screen pretty much at random. But as you look and see more and more of these impacts, you start to see this pattern of bright and dark bands appear. This is due to the interference between atoms acting in a wave-like way. And this wave-like behavior has even been seen for some quite large molecules containing dozens of atoms. And some researchers are aiming to show it for proteins, to show that whole protein molecules can also behave like waves. Now, there's no indication that that sort of wave-like property of molecules is relevant to biology, but that's not the point I want to make. This smeared-out character that atoms and subatomic particles can possess has some strange consequences. It means that in quantum theory, you can't speak of particles like this as having a definite position in space, but only of the probability that they will occupy a particular position in space. And this probability can, depending on the conditions, such as the way you look, can be quite tightly constrained, as it was in the atomic snapshots that I showed earlier, or it can be rather spread out. Even if you put an obstacle in the way of a quantum particle that should be unable to get past it, to get across to the other side, there's a small chance that it still might spread out and be found on the other side of an obstacle like this. In other words, the particle can seem to tunnel through this, what ought to be, impenetrable barrier. And in fact, that's how the earlier pictures that I showed you of atoms were made. They used an instrument called the scanning tunneling microscope, in which a very fine metal tip is brought close to a surface. And if there's an electrical voltage between the tip and the surface, there's a very small chance that electrons will tunnel across the gap between them. And if the tip and the sample are connected in an electrical circuit, this gives rise to a tunneling current, a flow of current because of the electron tunneling. And because that current is extremely dependent on the precise separation between the tip and the sample, any small obstacle on the surface, like a single atom just poking up from the surface, can produce a large change in that tunneling current. And in this way, the current measures the topography of the surface as the tip is scanned over it. Now, loosely speaking, the amount of quantum smeared outness of a particle, it can increase the greater, the smaller the particle is. So very small particles tunnel more readily than larger ones. Atoms can tunnel in principle, but the probability that they will do so is generally only significant if they're particularly tiny atoms. And the smallest, tiniest, lightest atom that there is the hydrogen atom. It has only one proton in its nucleus. Now, moving around hydrogen atoms from place to place is immensely important in many biochemical processes. There are several enzymes in our bodies that do just that. Here's a couple of them. They're involved in things like the generation of energy and in respiration. In general, the reactions involve moving a hydrogen atom, that's to say, in effect, a single proton, from one location where it's bound to perhaps one part of an atom or a molecule, moving it to another location. And that costs energy because the proton has to be transported over an energy barrier. It's a bit like plucking an apple off a tree. You have to tug at it to get it off. But it seems that in some, perhaps most processes of biochemical proton transfer like this, the process is made easier by tunneling. That's to say, the proton can tunnel across this gap, rather as if the apple on the tree suddenly vanished from the branch and just appeared in your hand. Now, how do we know that that happens? Well, as I say, the ease of quantum tunneling depends on the mass of the particle. The heavier it is, the less it tends to tunnel. So if we replace an ordinary hydrogen atom, in a case like this, with an atom of heavy hydrogen, the isotope deuterium, which has about double the mass of an ordinary hydrogen atom because it has a neutron in its nucleus as well, then we should alter the tunneling rate significantly. But at the same time, the chemical properties of the hydrogen are virtually unchanged here. Different isotopes of elements, which differ in the number of neutrons they have in their nuclei, are chemically identical. So all we change in this case is the mass, and therefore the tunneling rate. And when chemists first did this experiment in 1989 for an enzyme called alcohol dehydrogenase, they found that indeed the rate at which protons are transferred is different for deuterium in a way that fits with what we'd expect from a change in the tunneling rate. And that's something that's now been confirmed for several other enzymes. So tunneling makes biological proton transfer easier and faster. Does this mean that biology uses quantum tunneling? Well, it probably sounds as though that's just what I've said. But actually, tunneling is almost impossible to avoid in a situation like this. In fact, even how hydrogen atoms, or more strictly hydrogen ions, protons, move around between water molecules in ordinary liquid water, even that involves a degree of tunneling. So while you could argue that tunneling is useful for biology, it's not clear that enzymes have evolved to make use of it. They may be just stuck with this thing that happens whether they like it or not. Some biochemists, such as the Nobel laureate Ariel Warshel, believe that quantum tunneling is not only of rather minor importance in biology, because it only speeds things up typically by factors of maybe 2 to 10, which is not a lot for an enzyme, but also that it's just a side effect, that it's not something exploited by evolution. As one theoretical chemist has put it, tunneling is a fact of life, but life has no special effect on tunneling. Now, protons aren't the only particle to tunnel in enzyme reactions. Some of them involve transferring an electron from one molecule to another. For example, this is a crucial aspect of photosynthesis, in which an electron is kicked off a chlorophyll molecule by sunlight and passed along a chain of molecules in the chloroplast membrane. Electrons tunnel even more readily than protons because they're lighter, and it's been known for years now that tunneling is also significant in biological electron transfer too. But one of the most celebrated candidate systems for quantum biology involves electron tunneling in a much more unexpected manner. And the idea here is that tunneling within the receptor proteins involved in our sense of smell enables us to detect and identify molecules from the way they vibrate. So in the olfactory bulb in our noses where smell takes place, there are membranes studded with protein molecules that have cavities where the smelly molecules get lodged. Now, of course, these molecules aren't intrinsically smelly. They smell precisely because they can slot into the sites on the receptor proteins and trigger some nerve signal. That signaling process is quite well understood now, but what remains unclear is exactly how the odorant molecules like these, how they start the process off. The conventional idea has been that the odorant and the receptor are like a key and a lock with complementary shapes that fit each other. But it can't be quite as simple as that. For one thing, we can smell many more molecules than we have different types of receptor. But also, some molecules with very different shapes can smell similar. Here are two groups like that, while some molecules with almost identical shapes, like these two forms of the molecule carvone, can smell very different. In 1996, the scientist Luca Turin, who is a specialist on perfumes and in fact once set up his own company to advise the perfume industry, suggested that smell might not work primarily on the lock and key principle. Instead, He thinks that odorant receptors are a bit like vibration sensors, that they can smell the way molecules that they bind are vibrating. Molecules with very similar structures can have very different vibrations, while differently shaped molecules can have very similar frequencies of vibrations, just as a saxophone and a trombone can play the same note, even though they're very different shapes. So in effect, the idea is that the vibrations change the distance between the odorant and the walls of the cavity in which it sits, and thereby help electrons to jump the gap, to tunnel across the gap. And it's this electron current that the receptor then senses to trigger a smell response. Now, if Turing is right, it should be possible to distinguish an odorant molecule containing ordinary hydrogen atoms from one in which they've been replaced by heavy hydrogen, by deuterium. That's because the heavy hydrogen isotopes that have twice the mass vibrate more slowly, even though the molecule's shape is pretty much unchanged. And there's some evidence that this can happen. There's some evidence now that fruit flies can tell the difference. In 2011, a team in Greece collaborated with Turing on experiments on fruit flies, where they placed them in a T-shaped maze like this, with two attractive odors wafting from each passage, one of them just a deuterated form of the other. And they found that the flies preferred the ordinary stuff over the heavy stuff. The researchers could train these flies using mild electric shocks to actively avoid the deuterated odorant, which is something they could only learn if they could tell them apart in the first place. And what's more, flies trained to avoid one kind of deuterated molecule would also show an aversion to a quite different undeuteраted molecule that happened to have a similar spectrum of vibrations. Now, another team has since shown that bees, too, can be trained to discriminate between ordinary and deuterated odorants. There's no evidence yet that humans can tell the difference, although I have been told there's a group of chemists in Prague who are trying this out in their lab, and they do tell me that they can get used to smelling the distinction between the two. Now, unfortunately, none of this actually proves that Luca Turing's idea is right. For one thing, the doubling of the mass for ordinary and heavy hydrogen means that they're not quite as chemically identical as isotopes of other atoms are, where the mass ratio is smaller. So it could be that these creatures are actually detecting very small differences in chemical bonding rather than in vibrations. And even if they're not, I've been told by one of the leading specialists on fruit fly behavior and fruit fly olfaction, Scott Waddell at Oxford, that there could still be more conventional explanations of these sorts of experiments. In any event, some heavyweight supporters of the conventional theory of olfaction, including some Nobel laureates, are bitterly opposed to this vibrational idea, even though physicists and biophysicists have shown that it could work in principle. My own opinion, for what it's worth, is that whether it's right or not, it deserves to be taken seriously as a splendidly inventive and stimulating proposal. Now, as well as having electric charge, a negative charge, electrons possess another feature, a quantum mechanical feature called spin. Now, whereas electrical charge is a familiar property from classical physics, spin is purely a quantum phenomenon. The name is misleading. It doesn't mean that the electron is literally spinning, although sometimes that analogy is loosely made because spin is related to the electron's angular momentum, which in classical physics comes from an object's rotation. An electron spin can take two possible values, which can be imagined as the electron spinning clockwise and anti-clockwise. And electron spin was first introduced into quantum theory in the mid-1920s by Wolfgang Pauli. And he based his ideas on theories that had just been formulated by Schrödinger and Heisenberg. And this led Pauli to propose the so-called exclusion principle, which states that no two electrons can share the same quantum state. And it's because two electrons can have opposite spin that two of them can share the same orbital in an atom. And this picture of how electrons share orbitals can explain the whole of the periodic table of the elements in terms of the arrangements of electrons, how the electrons fill up the available orbitals. So this idea of spin is central to understanding the whole of chemistry. And because of their spin, electrons are magnetic. You might again crudely think of this as being a little similar to the way the spinning Earth has north and south magnetic poles. For this reason, the spin states are often referred to as spin up and spin down, although again you shouldn't take those labels too literally. One way to flip the spin of an electron from up to down, say, is to put it in a magnetic field, which can realign these poles. Now, in recent years, there's been a lot of interest in constructing a form of electronic technology that makes use of electron spin. In ordinary electrical currents, the electrons have a mixture of these two spins, and it's not an important factor at all. But if we could control the spin states of electrons, say using magnetic fields, then we might be able to use spin as another variable for encoding binary information and to carry out digital logic like that used in computer circuits in a new way. And this idea is called Spintronics, and it depends on making electronic devices that can do things like allow electrical currents to pass depending on the electron's spin. Now, this is hard to do because it's very hard to stop the spins from getting randomized, and so far we really only have a handful of spintronic devices. At least one of them is hugely important, though, and because the magnetic effects on electron spin can create an effect called giant magnetoresistance, where the electrical conductivity of a device becomes highly sensitive to the presence of magnetic fields. And devices that use this giant magnetoresistance are used in the readout heads of computer hard drives to read the information off the magnetic disk. But it seems that biology already knows how to do spintronics. Ron Naaman at the Weizmann Institute in Israel and his co-workers have found that various biological molecules and systems, such as some of the membranes involved in photosynthesis, can conduct electrons selectively depending on their spin. This is something that DNA, in fact, can do also. It's not completely clear whether this happens in real organisms, or if it does, whether it serves any role in biology. But it could be useful for spintronic technology. There are some proposals for using biological molecules or tricks learnt from them in these devices. What's more, Luca Turin and co-workers have recently presented evidence that anesthetics, whose molecular mechanism of action has long been a mystery, might act by changing electron spins so as to enable electrical signaling between certain kinds of protein molecules. But there's an even more striking way in which electron spin might feature in biology. The fact that spin makes electrons magnetic has long been thought to account for the fact that some chemical reactions can be sensitive to magnetic fields. You see, electrons generally tend to pair up in orbitals. That lowers their energy, and that's how chemical bonds are formed, by the pairing up of electrons on different atoms. And these electron pairs in a single orbital have opposite spins. That's Pauli's exclusion principle. And so the pair has no net magnetism in that case. They cancel out. But some reactions, including some important ones in biology, involve the splitting apart of chemical bonds to form pairs of atoms or molecules that each has a lone unpaired electron. And those molecules are called radicals. And because they have a net electron spin, they feel magnetic fields. And this means that the reactions can be sensitive to magnetic fields. Radicals like this are generally very reactive. They don't stick around long before they find some other atom or molecule to combine with. Now, for many years, it's been suspected that this might help to explain the magnetic compass that some birds seem to possess, which allows them to migrate by using the Earth's magnetic field. Lots of birds do this. Robins do, some sparrows do, and homing pigeons. And the idea is that the geomagnetic field, despite being so weak, somehow influences the rate of some biochemical process in the bird's brain, so that the neural signaling differs depending on how the bird is oriented in the geomagnetic field. And this seems to be very probably what's going on. What's more controversial is a recent idea that this avian magnetic compass uses perhaps the most bizarre effect in quantum physics, which is called entanglement. Now, Erwin Schrödinger, who gave entanglement its name, proclaimed that it's actually the central concept of quantum theory. That he said quantum theory isn't so much really about quantum jumps or wave-particle duality or particles being in many states at once. It's about entanglement. And the irony is that the possibility of entanglement was first identified by the man who, having more or less started quantum theory in the first place, by the 1930s had become one of its greatest skeptics. That's Albert Einstein. Now, it's not that Einstein thought the quantum theory was wrong. He just thought that it was incomplete. According to the idea of Niels Bohr and Werner Heisenberg and their colleagues in Copenhagen in the 1920s, quantum theory imposes fundamental limits on what we can know about the world. And in their so-called Copenhagen interpretation, all you can know is what experiments tell you. So the fact that experiments seem to insist on some uncertainty about quantum states, or that different experiments might tell you different things, wasn't because of some limitation in what you could measure. It was because there was literally no deeper truth beyond that to be found. So when quantum mechanics failed to predict, say, whether an electron would have one spin or the other until you measured it, this wasn't because the theory was incomplete, but because there was simply no meaning to that question before you had made the measurement. So this is the Copenhagen interpretation, and it's what Einstein couldn't accept. He was convinced that there had to be a deeper truth, what became called hidden variables, which assigned everything with a definite one or the other value, even if we couldn't find out what it was. And with two younger colleagues, in 1935, Einstein published a paper showing why, in his view, the Copenhagen interpretation had to be wrong. He imagined an experiment in which two particles would be generated with quantum states that had to be related to one another. Let's say a pair of electrons in which the spins would be opposite, one spin up and one spin down. And the idea is that the electrons would be spat out in opposite directions from some device. Now, we don't know which of them has which spin until we measure it. Whereupon we know, so we make a measurement on one of them when we figure it has that spin, and so we know that the other one must have the opposite spin. And it's because of this correlation between spins that we know that this must be the case, and that in that sense, this pair of electrons is said to be entangled. Now, if the Copenhagen interpretation is right, then it's not just that we don't know which spin is which until we make the measurement. It's actually that the spins are literally undecided until then. And when we make the measurement, that decision is made by the universe at random. By making the measurement on one electron, we force the choice to be made. But then it would seem that that choice has to be communicated instantly to the other, even if they've become separated by vast distances. Einstein called this spooky action at a distance. And he said, how can this information be sent instantly across such a distance? In fact, that idea seemed to violate his theory of special relativity, which said that no signal can travel faster than light. Now, in his view, there was no need for something as weird as this, because he said the spin states must have been assigned to the electrons all along. It was just that they were somehow hidden from us. his challenge to the Copenhagen interpretation stimulated lots of discussion, but there was no obvious way at that time to test it. And it wasn't until the 1960s that the Irish physicist John Bell proposed an experimental way to do that. I don't have time to explain what Bell proposed, but his theory was put to the test in the 1980s by Alan Aspect in France. He's got this splendid moustache which sort of invites jokes about entanglement that I won't try and get into. He made measurements on entangled pairs of photons coming out of a laser, and he measured not spin states, but their states of polarization, whether they were horizontal or vertical. And these experiments showed that there can be no hidden variables after all, that entangled photons really behave exactly as if spooky action at a distance is real. Now, I need to qualify that statement. First of all, there are various loopholes in the argument that excludes hidden variables from this picture, and that they've only been very recently closed off. Second, entanglement doesn't actually violate special relativity, because it can be shown that although there seems to be this instant correlation between the particle states, its interrelation, we can't establish that in any way that, that transmits the information faster than light. And third, this doesn't amount to saying that this spooky action at a distance is real. It only looks that way if we're thinking in terms of Einstein's idea, in which particles really do have locally self-contained states. What entanglement really shows is that quantum theory involves so-called non-local effects, meaning that there are situations like this where we can't think of what is happening here as being independent from what is happening here. It's a non-local interaction between them. Okay, back to the birds. This magnetic sensor, then, that they have is activated by light striking the bird's retina. And the best guess at the mechanism for how it happens seems to be that the energy deposited by each incoming photon creates a pair of these radicals, in which the unpaired electrons that they contain both have the same spin. And as the radicals move apart, they experience different magnetic fields. They both feel the Earth's magnetic field, but maybe one of them also feels a magnetic field from a nearby magnetic atom. And the difference in the fields shifts the radical pair between two quantum states with different chemical reactivity, perhaps some signaling chemical synthesized in the bird's retina. It happens at a different rate when it's in these different states. And the relative balance of those states would then depend on the orientation of the bird relative to the Earth's magnetic field. So that's the general idea. Simon Benjamin in Oxford and his co-workers have proposed that these two unpaired electrons, being created by the absorption of a single photon, have spins that are in a state of quantum entanglement. And although entanglement is usually quite delicate at ambient, at room temperatures, the researchers calculate that it could be maintained in this avian compass for at least tens of microseconds, which is considerably longer than it's usually possible to sustain it in artificial systems in the lab. They argue that this entanglement is essential for the extreme sensitivity of this system to very small magnetic fields, because magnetic field effects experienced by one of the radicals can be, in some sense, felt by the other one some distance away. Well, that's all very interesting, but it won't be proved unless we can identify what the radical pairs involved are, at least if we can't show that. Magnetoreception in birds, and also in some other magnetic field sensing organisms like fruit flies, seems to be mediated by a kind of protein called a cryptochrome, which will produce radical pairs. No one is quite sure how this happens in detail. Recent experiments in flies have pretty much established that this is the sort of conventional idea, and you see the shifting around of electrons here that essentially creates radical pairs. This is the conventional idea for what's going on, but it seems that recent experiments have ruled out this particular pair of radicals as the ones that can be involved. One of the experts in this field, Steve Reppert at the University of Massachusetts Medical School, tells me that any explanation for this process is going to have to come up with a different way to generate these radical pairs. So there's still a lot to be understood here. Now, I talked earlier about how quantum effects seem to depend on size. They might appear in very small things like atoms, but not in big things like us. No one yet knows whether that's a fundamental fact or not. That's to say, the reason footballs can't tunnel might not be because they're big, but rather because they're so complicated, because they're made up of so many atoms. Each atom has a wave-like nature, but the waves all get jumbled up and out of step, so that these wave properties get averaged away, like soldiers marching out of step. The technical term for this jumbling is decoherence, and according to one view, the reason why quantum behavior at small scales becomes classical behavior at big scales is because of decoherence. And so this is a relative thing. The more atoms an object contains, the more decoherence kicks in, and the less likely it is that we'll see these quantum effects. If we want to make use of certain quantum effects, then we generally have to control and suppress this process of decoherence. One way to do that is to isolate a quantum system from its environment. The jostling that it would receive from all the atoms around it is something that would cause this decoherence. Now, an area where this is important is quantum computing. And here, the aim is to encode information in quantum states, just as we currently encode information in, say, in magnetic domains on a magnetic disk, or in arrays of light-scattering dimples on a CD or a DVD. The advantage of a quantum computer is that a single quantum bit could be placed in several states at once, not just 1 or 0, but a bit of both. And this is a so-called superposition state. This means that quantum bits could, in effect, perform many calculations at once. At least that's the simplistic explanation. It's the one I'm going to give tonight. The truth is that actually no one is yet really sure where the sort of speed up that quantum computers are thought to offer, quite where it comes from. But it's really hard to do this because these coherent states of quantum bits are generally delicate and easily disturbed. They're very vulnerable to decoherence. And that's why, so far, we have quantum computers with only a handful of bits, and why, generally, they need to be cooled to very low temperatures. Even the one quantum computer currently available commercially, this one made by the Canadian company D-Wave, it looks like one of the old mainframe computers. It's the size of a room, and that's because of all the cryogenics that is needed to keep the quantum bits cool. I should say that that there's a real debate at the moment about whether D-Wave's quantum computer really uses quantum effects or not, but that's another matter. So you can imagine then, perhaps, why people were so astounded by a claim made in 2010 that coherent quantum superpositions of energy states can be created and sustained in bacteria and algae, and perhaps in all green plants that use photosynthesis. And that this is what makes photosynthesis, which is really the font of nearly all life on Earth, what makes it possible. Photosynthesis starts when a chlorophyll pigment molecule absorbs sunlight, and that energy is then transmitted through a forest of pigment molecules around it until it reaches the so-called photosynthetic reaction center, and there it causes an electron to be spat out and sent down a relay of molecules where eventually it's used to split water and produce the energy-storing molecules of the cell. And the standard view was that this energy, bundled into wave-like packets called excitons that are spread out over several pigment molecules, that this hops at random between the pigment molecules until it finds its way to the photosynthetic reaction center. And as you can imagine, finding this path by random hopping is pretty inefficient and it's time-consuming, during which some of that captured energy may be wasted as heat. But researchers in America reported experimental evidence that they said showed that photosynthetic bacteria and algae contain excitons that move not in these random hops, but as coherent waves of quantum superposition states. If the exitons are in this coherent superposition, then they can explore many paths at once, and this should allow them to find the best route much more efficiently and quickly. It was suggested that this was, in effect, the same as the way a quantum computer would perform many calculations at once, that, in effect, photosynthesis is a kind of quantum computation. Which sounds pretty amazing, right? But is that really how things are? Well, since those experiments were conducted 5 years ago, there's been a lot of debate about the interpretation, and some researchers now think that the results originally interpreted as quantum coherence might have other explanations, in particular that they might be caused by more conventional interactions between the excitons and the vibrational states of the pigment molecules. While we need some quantum theory to understand that process in any event, it's not at all clear that what the researchers are calling coherence here is indeed the same as the quantum coherence that is being sought in quantum computing. So, in fact, we still don't yet really know if photosynthesis truly is a manifestation of quantum biology. And even if it is, we have again to ask the question, has biology adapted to make use of this effect? Or is it inevitable? It's all too tempting to attribute it to the wonderful power of natural selection to find good solutions to problems. But we can't simply assume that that's what's happened. It might be that if you need to pack together pigment molecules densely in an array to get to maximize light absorption, something like this coherence of exciton states is unavoidable, that it's just a byproduct, not an adaptation. After all, most photosynthetic organisms are spoiled for light. They don't need to worry about conserving every last bit of photon energy, and so there's not obviously any selective pressure to get them to make use of quantum tricks like this. Well then, I hope you can see that there are some extraordinary possibilities in quantum biology, but also many uncertainties about how far it reaches. Nearly all of the prize candidate systems in quantum biology still have plenty of questions to answer. But some speculations about quantum effects in life go much further, and I want to end by just mentioning a couple of them. One of the biggest questions facing the life sciences today is how the brain works, and in particular, the origin of human consciousness. In his 1989 book, The Emperor's New Mind, Roger Penrose suggested that consciousness may have something to do with quantum theory. He suggested that the choice that seems to happen when we make a measurement on a quantum system, a choice called wave function collapse, because it collapses all the possibilities into just a single outcome, that this might underlie processes that are non-computable, ones that a computer can't calculate or simulate. And he suggested that the human mind is capable of dealing with processes like this, and that wave function collapse might therefore be important in some way for thinking and for consciousness. And in conjunction with the physician Stuart Hameroff, he later suggested that entanglement between electrons on protein structures called microtubules in the brain could be at the root of this process. Now, like many, I should really say, of Penrose's wilder ideas, this one is tremendously inventive. And like many of them, there's also not a shred of current evidence to support it. But some interpretations of quantum theory imply far, far more profound consequences for biology than any of this. The collapse of the wave function is in fact a very puzzling thing, because there's nothing in conventional quantum theory that explains or predicts it. It's something that has to be put into the maths by hand. And some physicists are very dissatisfied with that. They think it's much more parsimonious to assume that in fact there is no collapse of the wave function at all. Instead, they say, all the possibilities contained in the equations of quantum theory correspond to real situations. None of them goes away when we make a measurement. Instead, the world, meaning really the entire universe, splits into alternative worlds when this happens, and that they're all real. but that our consciousness can only be present in any one of them. So you could say in one of these worlds, Schrödinger's famous cat survived, in the other one it died. And both happened. This is the so-called many worlds interpretation of quantum theory. It was first proposed in the 1950s, and it's now advocated by some of the most influential physicists in the world, including Stephen Hawking and Frank Wilczek, as well as well-known popularizers like Brian Cox and Max Tegmark and Brian Greene. In this view, quantum physics offers you another way to replicate. It's a whole lot less fun than the conventional one, but you can't avoid doing it more times a second than you can possibly imagine. Every time a quantum event occurs anywhere in the vicinity, perhaps anywhere in the universe, you split into multiple selves. Well, I felt obliged to mention this idea. My own view, which I don't have time, fortunately, to justify, is that this idea richly merits Wolfgang Pauli's famous put down, "It's not even wrong." In another universe, I'm going to dash out right now and go to the pub. In this universe, I'm happy to stay and answer questions. Thank you very much.
Does the cooler temperatures just slow it down so that we can observe it more, or do they simply not work? Because in a quantum computer, would they not work at sort of higher temperatures?